In this lesson, we'll continue our discussion of the four important biomolecules by taking a look at lipids. So, like carbohydrates, lipids contain carbon, hydrogen, and oxygen. If you remember, carbohydrates were for every one carbon, there were two hydrogens, and then one oxygen. That's not the case here. Lipids don't have quite as much oxygen as carbohydrates, so they contain less oxygen, and the ratio of carbon to hydrogen is 1 to 2. For every one carbon, there's two hydrogens, and I'll show you that on some diagrams. Now, some lipids do contain phosphorus. We're going to see as an example something known as phospholipids, and they're very important in our cell membrane structure. Most lipids are hydrophobic, meaning that they don't like water, right? So they're typically insoluble in water. Oil would be an example of a lipid, right? So if you took a droplet of oil and you put it into water, we know that oil will not mix because it is hydrophobic. So we see lipids in a bunch of different functions. Some of the important ones we're going to look at this semester are going to be it forms the structure of cells, specifically the cell membrane. I'm going to go into this in great detail when we get to cell structure in subsequent lessons. We also use lipids as our energy reserve, right? When we have more, say, calories than we burn, we could take those calories, let's say sugars, and we can convert them into lipids and store that fat for a time when we need that energy. So there's a few classes of lipids we're going to look at. We're going to look at fatty acids. We're going to just mention icosanoids. This is something that's a little bit advanced. We'll then take a look at glycerides. These are important to us. These are our body fats. We'll take a look at steroids. These become important as you go on in your studies in A&P2. You'll see different hormones and things like that are steroids. This semester we'll look at cholesterol. Actually, that's a part of our cell membrane, is a steroid also. Phospholipids, very important in cell membrane structure, as well as glycolipids. We're going to focus mostly this semester on phospholipids. If you ever go on in your studies, you'll see how important glycolipids are in cell membrane structure also. Okay, so an important slide, lipids contain carbon, hydrogen, and oxygen. Again, the ratio of carbon to hydrogen is 1 to 2 with not quite as much oxygen. Again, most lipids are hydrophobic, right? That means that fat does not mix with water, and we're going to see some important functions are going to be cell membrane structure and energy reserves, right? It's our stored form of energy. So let's take a look at the fatty acids first. Right, these are actually quite large. These are long chains of carbon, and then you're going to see a hydrogen on each carbon and hydrogens, I should say, on each carbon. And at the very end, the functional group, which is an organic acid known as a carboxyl group. Let's take a look. So if you notice over here, we have these carbons. Watch this. When you first see this, it looks a little bit intimidating. It's not that bad. Just follow this central, I call it like a backbone here. All these carbons form a chain. So if we follow my cursor here, they form a chain, or that whole chain of carbons. To me, that's the backbone. Then take a look, guys. Off of each carbon is one, two hydrogens. One, two hydrogens off of there, right? Here's your carbon. One, two. Carbon, right? One, two carbon. One, two. Now this one has three because there's no other carbons attached to it. So this is a long chain of carbon. Then at the end over here is our organic acid. This is the carboxyl group. If you remember from our previous lesson, C double bond O, OH. That's your carboxyl group over here. All right, this is just a structural rendering of this. Each black area here would be a carbon, and then the blue is going to be the hydrogens. I'm not too concerned about that. Okay. Um, this is, and the red is going to be oxygen if you're interested. This is the picture that kind of tells it nicely for us. Now, These are relatively non-polar, right? Meaning that they don't mix, that they don't have a charge on them. So as a result, they don't mix with water. The only part that may have a little bit of a charge is the carboxyl group, this over here, right? This would interact with water, but there's so many carbons here that are equally distributed between carbon and hydrogen. There's no charge to this structure here. So we say this whole molecule is non-polar. It doesn't have a charge. So that's the reason it doesn't mix with water. If you remember from previous lessons, non-polar structures do not interact with water. They are hydrophobic. Now, we can classify fatty acids as either being saturated or unsaturated. Very simple. If all the carbons are attached to hydrogen, and if you look at each one of these carbons here in the main chain, don't worry about the carboxyl group. We're not talking about the functional group. But if you look at every carbon in the backbone here, each one is attached to hydrogen. We can't add any more hydrogen because each carbon is covalently bonding four bonds. If you take a look at each carbon, you'll notice it has four lines attached to it. One, two, and then three, four between the carbons. So each carbon has four bonds. We had gone over that in a previous lesson also. So we can't add any more hydrogen to this structure. So we call this a saturated fatty acid. Okay? Saturated means that it is filled with hydrogens. And you'll notice that saturated fatty acids only contain single bonds. These we typically see more in like animal fats, right, in meat and things like that. They're typically solid at room temperature and generally more on the unhealthy side. Right? People that eat a lot of saturated fatty acids are more prone to like heart disease and that type of thing. If Let's go here. Let's say one, two, three. Let's go here between the fourth carbon and the fifth carbon. Let's just say this hydrogen was gone and this hydrogen was gone here. That means we could add more hydrogen. That would be an unsaturated fatty acid. But here's the deal. There's an electron that's being shared with this hydrogen from carbon, and then there's an electron being shared with this hydrogen. That means those two electrons wouldn't be shared with the hydrogens. They would have to share with one another, and that would force what we call a double bond. So with unsaturated fatty acids, where the hydrogen is missing, which would be here, we're typically going to see two lines is going to be the sharing of two pair of electrons. And that's how you can tell you're looking at an unsaturated fatty acid. It is going to have a double bond. Now, typically, unsaturated fatty acids are healthier for us. This is what we find in plants. Your oils, like olive oil, right? They're going to be predominantly unsaturated fatty acids. So we do have these as part of our structure also. But to an unsaturated fatty acid, we could add hydrogens and turn it into a saturated fatty acid. You'll also see unsaturated fatty acids typically are bent. I'll talk more about that in subsequent lessons when we look at the structure of cell membranes. So an unsaturated fatty acid has one or more double bonds. Okay. If there's just one double bond, we call that a monounsaturated fatty acid. If there are many, we call them polyunsaturated. Okay, so there's a combination of these different saturated and unsaturated. But in nature, the unsaturated fatty acids, like I said, they're more of our oils that we see plants typically. Like I said, olive oil is a good example. Okay, so if you look at this real quick, you can see it's all single bonds, saturated fatty acid. If you see a double bond along the chain, unsaturated. If it's just one double bond, mono unsaturated. If there would be like one here, and then one down a little bit further, that would be poly. Okay, so those are our fatty acids. We'll look at them again, and when we look at glycerides, actually, was in a couple minutes. The second category of fats or lipids are the icosanoids. So this is a little bit advanced. you're going to study these more in like an A&P2 class or a pathology class, that type of thing, or an internal medicine class, something like that. These are derived from a specific fatty acid known as arachidonic acid. Now, we don't get this in our body by itself. We actually have to ingest it from the diet, right? So it's not synthesized, it's not made in the body. We have to obtain the arachidonic acid from our diet, from the foods that we eat. Now, there's two classes of icosanoids. Leukotrienes are the first one. These are actually what help to cause inflammation. When you have inflammation, these chemicals are released to cause inflammation, right? So they typically respond to some injury or disease. Don't write this down, but a lot of times asthmatics, people that have asthma, produce too much leukotrienes, and they'll actually take drugs that are leukotriene inhibitors, right? They actually block the release of leukotrienes. Another group, which you're going to see more in AMP2, are known as prostaglandins. They do so many things for us. Many times they're considered just local hormones. They kind of work in the area of the cells that are releasing them. So again, this is a little bit advanced. Just know that icosanoids are derived from arachidonic acid. Then there's two categories or types of icosanoids: leukotrienes, which cause inflammation, and then prostaglandins, which kind of just function as local hormones. They just do so many different things, way beyond the scope of where we are at this point. All right, that you'll get into more in like an A and P2 class, or even in a pathology class. So just to show you, don't have to know this, just to kind of give you an idea. Here's a prostaglandin. So you can see here, this is 5 carbon ring right here, right? 1, 2, 3, 4, 5. And then attached to it is a fatty acid chain and a fatty acid chain. So if I was in class with you, I would ask you, this fatty acid chain right here, is this saturated or unsaturated? How about this one? Saturated or unsaturated? And then since you see the double bond here and a double bond here, both of these are unsaturated. And so we have two unsaturated fatty acid chains attached to this 5 carbon ring. You do not have to know the structure. I just thought I'd show it to you. Okay, let's take a look at glycerides. This is going to be the third class of lipids. This one's important to us because this is what's going to make up our body fat, right? So this consists of fatty acids, right? We're going to see our fatty acids either saturated or unsaturated attached to an alcohol. The name of this alcohol, give this a little highlight, is called glycerol. This is an alcohol. Now, if I take glycerol and I just add one fatty acid to it, I would have something known as a monoglyceride, right? Mono meaning one, right? So I have the glycerol and I just add one fatty acid to it, we would call that a monoglyceride. If I take that and I add another fatty acid to it, right? Take the monoglyceride and now add another fatty acid so that I have two fatty acids attached to the glyceride, that would be known as a diglyceride. But the one that's important to us in the human anatomy and physiology are the triglycerides. This is going to be an alcohol, glycerol, with three fatty acids. And this, my friends, is our body fat. Okay? And I'm sure you've had blood tests, or you know people that have had blood tests where they check their triglyceride levels. Now, a biochemist would call them triacylglycerols, or some people just call them neutral fats. For our course, people that are going into medicine, right, most of us call it triglycerides. So why do we have these in our body? They are an important energy source. We can actually break down fats for energy. We know also that it acts as an insulator. It helps to keep us warm, right? You look at animals that live in cold environments in the Arctic or Antarctic, right? They have layers of blubber on them, right? For not only energy but for warmth. And then triglycerides, our body fats, are found underneath our skin, which helps to protect our bones and things like that, the muscles underneath them, but also a lot of organs like the kidney is a great example, is packed in fat. Our heart has fat on it. Our intestines have fat on it. So it's a protector. Okay, so the important one we're going to look at, guys, is going to be the triglycerides, which is the alcohol glycerol plus 3 fatty acids. So again, when you look at this, you're like, oh my goodness, this looks a little bit intimidating. Watch this. In purple, this structure here, it's 3 carbons with an OH group attached to it here, here, and here. OHs are make something an alcohol. So this whole purple box right here represents glycerol. Now, if I take a fatty acid, how do I know this is a fatty acid? It's a long chain of carbons. Here's your carboxyl group, C double bond O, OH, right? So that's a fatty acid. If I take this fatty acid, guys, and I attach it to the glycerol, what do I remove? H2O. Again, if we were in class right now, I would say, what chemical process is this where we remove a molecule of water to form a bond? And that you're going to say, and I know you would answer this correctly, you would have said dehydration synthesis. So watch this. When I take this water out, I'm going to put it over here, and now look at this. I have a bond right here. Don't write this down, but for those of you that may have taken chemistry before, this is known as an ester bond. All right, you don't have to know that from me, just to throw it out there in case you've ever heard it before. So this was a saturated fatty acid. How do I know it's saturated besides that they wrote it there? How many double bonds are there? None in the main chain, right? We're not talking about the carboxyl group. We're always just talking about the main chain. So if I just attach this one fatty acid to this glycerol, that would be a monosaccharide. Now, I take a second fatty acid that happens to be also saturated, right? No double bonds. And same thing, we take out a molecule of water. I'll take the OH from the carboxyl group and this hydrogen from the hydroxyl group over here. Take out that. I'm going to put it over here. And now I have a bond between this carbon and this oxygen. And now we have, if I just had these 2 fatty acids here, 1 and 2, that would be a diglyceride. But when I add this third fatty acid on here, if you notice this has a double bond, so that's unsaturated. Same thing, I take out that third water molecule, I form the bond between carbon and oxygen. There's the bond right there. Third water. Now this whole structure, guys, here's your glycerol. 1, 2, 3. Triglyceride. Right? This is our body fat. This is what we make and we store in our fat cells. You're going to learn this in a subsequent lesson. We call them adipocytes. Right? These are cells that store fats. So notice when we went this way, we took the glycerol plus the 3 separate fatty acids. Going this way is dehydration synthesis. But how about this? What if you consume this? Let's just say you had some meat and there was fat on it, and we're going to now digest it. We're going to break it down. We can add the water back in, right? These 3 waters, add them back in, and there's certain enzymes with that now go this way where we add the water back in. It remember is hydrolysis, right? Hydrolysis. Hydro is water. Lysis means to break down. Okay, so you see when we go this way, it's hydrolysis. Okay, let me just go back to make sure we got everything. All right, so monoglyceride again is a glycerol plus 1 fatty acid. Diglyceride is a glycerol plus 2 fatty acids. Triglyceride, that's the one we're going to work with mostly this semester and in A&P2, is a glycerol plus 3 fatty acids. And again, all of this is created by dehydration synthesis, where we remove a molecule of water. 1, 2, 3, right? 1 per fatty acid that we attach to the glycerol. Okay, so we did our fatty acids, we did our icosanoids, we did our glycerides, which are our fats. The 4th category of lipids that we want to look at, we just want to introduce them this semester. And excuse me, the most of them we're going to look at more next semester. We will look at one called cholesterol. These are much bigger structures. These are rings of carbon where there's actually 4 rings. Watch this. 1, 2, 3, 4. At each corner, guys, we don't put it there, is a carbon. Right, so at every corner is a carbon. So look how big this is, right? Here, this happens to be cholesterol. You may have heard of estrogen and males, testosterone. These are all steroids. Okay, so these are four ring structures, and then they have different functional groups that give them different properties. So the important one for us this semester is going to be cholesterol. We're actually going to see when we get to cell anatomy, cellular anatomy, cholesterol is part of our cell membrane, the plasma membrane. So we will look at that this semester. In subsequent lessons in A&P2, you'll take a look at some of the sex hormones, estrogen, testosterone, those are steroids. Different corticosteroids, these are stress hormones. You don't have to write this down, but if you've ever heard of cortisol, that's a corticosteroid that comes from our adrenal gland. That's a steroid. Calcitriol comes from our kidney for calcium regulation. Bile, if you've ever heard of bile, that comes from our liver. It's stored in our gallbladder. Bile has bile salts that are steroids. These are necessary for the digestion of fats. When we digest fats, we need these bile salts. So these are things here, the last three dots, we're going to see more in A&P2, where we talk about hormones and we talk about digestion and reproduction and that type of thing. This semester, we're going to focus on cholesterol. You do not need to know this slide, right? I would never ask you to identify cholesterol from estrogen to testosterone. I just wanted to give you some examples and just show you what they look like. But steroids are very important to our anatomy and physiology. The last two class, I guess, of lipids is going to be phospholipids and glycolipids. I'm going to combine these together because both of these are an important component of the plasma membrane. If you don't know what that is, that's the cell membrane. Okay, these we do make in our body, and we'll talk about where they're made in subsequent lessons. Now, these are contain a diglyceride, so they're going to have two fatty acids attached to either a phosphate group in a phospholipid or a sugar in a glycolipid. Whenever you see glyco, think carbohydrate, believe it or not. So let me show you what I mean. So again, when you look at this, you go like, oh my goodness, this is a little bit intimidating. But watch this, guys. Here's our diglyceride. See the purple box right here? That was the glycerol. And then here's one fatty acid, right? If you look at it quickly, and again, if I was in class, I would ask you this, is this saturated or unsaturated? And if you see the double bond right there, unsaturated. And also unsaturated fatty acids are usually bent. Then the other fatty acid is a straight chain with all single bonds. So this would be a saturated fatty acid. Right? So we have a glycerol attached to two fatty acids. So this would be the diglyceride. Then up over here, guys, watch this. Excuse me, is the phosphate group right here. So this is called a phospholipid. Again, we're going to see this is an important structure in our cell membrane. I'm going to throw this out here now. This top part here is charged. So this would be hydrophilic. We know that fatty acids are hydrophobic. So this part of a phospholipid we're going to see in the subsequent lessons is going to face water, where this part down here, where the fatty acids are not going to face water. Okay, so this is an example of a phospholipid. The glycolipid, again, is going to be a diglyceride, but instead of having a phosphate head, the top part, it's going to have a sugar. What do I mean by that? So watch this. Glycerol, and then again, one fatty acid, second fatty acid. That's the same. All right, so that's the diglyceride part. But notice up here, this top part doesn't have phosphate group. Instead, it has, hopefully you recognize that, kind of looking like a sugar, like glucose, right? So it's going to have carbohydrates up on top. And this is going to be known as a glycolipid right there, glycolipid. All right, these two, again, we're going to look at. I just want to introduce them to you today, let you see what they look like. And then when we get to subsequent lessons on how we make the cell membrane, I will remind you about these. So generally, both of these have hydrophilic heads. That's the top part, but hydrophobic tails. Again, what do I mean by that? This top part here, right here, sorry, this top part is the head, and then these down here are the tails. The top part is hydrophilic, likes water. The bottom part, hydrophobic, doesn't like water. The top part here is hydrophilic. Again, same thing, the bottom part here is hydrophobic. Now, here's a simple way. If I was in class, I would actually draw these on the chalkboard or the whiteboard. I would draw them like this. It's a hell of a lot easier, right? This represents the phosphate head and then the two tails, right? So again, we're going to see this part here is hydrophilic. It likes water. And the tail is hydrophobic. And then this would be the glycolipid. Head, tail. Again, the only reason I'm going over this now is we're going to need to know this in a future lesson. Okay? The hydrophilic heads, again, are attracted to water. Hydrophobic tails are going to stay away from water. And just the last slide. So same as we had with carbohydrates, again, these textbooks do a great job with this, where they just kind of do a summary table where they show you the lipid type. They give you some examples, again, some of their major functions, and then any other remarks. Okay, so that concludes this lesson on the second biomolecule that we're studying, which are the lipids.